Now our next speaker is Professor Swapandeep Singh Chimni. His brief about his biodata. Swapandeep Singh Chimni received a BSc Honours 1984, MSc Honours 1985, and PhD 1991 degrees in chemistry from Guru Nanak Dev University, Amritsar. He is currently holding the position of Professor of Chemistry at Guru Nanak Dev University, Amritsar. From 1990 to 1992, he has lectured in applied chemistry at NIT Jalandhar, where he initiated his research on biocatalysis from 1992 to 1997. He was a lecturer in chemistry at GNDU Amritsar and was promoted as senior lecturer in 1997, subsequent as reader in 2000. In 2009, he was promoted as Professor of Chemistry. His research interests include enantioselective synthesis of small molecules using biocatalytic and organocatalytic methods. He has authored more than 124 publications with his students and collaborators. Two of his publications received most cited paper awards from Tetrahedron Letters 205 to 208 and Tetrahedron Asymmetry 205 to 2008, 6 to 9. His two research papers were highlighted as Insight Cover 2013 and 14 in Asian Journal of Organic Chemistry, and another two highlighted in Chemistry Views 2012-13. One of his research papers has been highlighted on the front cover of Organic and Biomolecular Chemistry 2015. Professor Chimni was awarded INSA RSC Bilateral Exchange Fellowship in 1996 to visit the laboratory of Professor Stanley M. Robert Robinson Laboratory, University of Liverpool, UK. He also visited the laboratory of Professor D. Basvaiya, School of Chemistry, Central University, Hyderabad, as UGC Visiting Fellow 2002-2003. He visited Institute of Organic Chemistry, Warsaw, Poland, under PSINSA Bilateral Exchange of Scientists in 2017, and delivered seven lectures in different institutes, universities in Poland. He has delivered over 27 invited talks in national and international conferences and gave a keynote address in a national conference at DJUSNT, Hisar, and has chaired sessions in many conferences, symposiums. He has bestowed with Postal and Chemical Research Society Award for the year 2014. In 2019, he participated in the Leadership for Academicians program of MHRD, Government of India, and visited University of Pennsylvania, USA. So, Professor Chimni sir, are you available here? Please continue your session.
Hello, thank you, Naseeb,
Welcome, sir,
for your nice words of introduction. So I'll just now be starting with what I intend to talk about today. So as we have just now heard Dr. Yadav, and he has been talking about, he started with green chemistry, and he's talked about the status of Indian science in a whole, and that to chemical sciences regarding energy generation at the end. Okay. So what probably he was, as he initiated his lecture, he was referring to something that in your daily life, chemicals are involved. From anything that you start when you get up in the morning and do your daily chores and all that. But well, obviously, because we all are breathing oxygen. Oxygen is a chemical. So if we don't breathe oxygen, we are no more. And this generates so many chemical reaction inside our bodies, which results in all those things. So it's very important that we study nature very closely and try to understand how nature is doing chemistry. And if we can understand how nature is doing chemistry, then obviously we can do certain man-made chemistry. We can just copy or mimic, as we call it, and then try to do those chemistry which nature is doing in the living organisms in the lab, in the flask that we work with normally. Okay. So that is the basic premise or the idea behind what we should be learning from. As my title was related with one of my projects which I handled with my students. It was exploring organocatalytic enantioselective carbon bond formation via decarboxylative addition reactions. But since I found that your conference has a bit more time for me, so I have included certain other things also. And I'll take you on a journey of how we got into this type of work and how we try to copy nature. That's what Dr. Yadav also probably was hinting at. So I'll first introduce to you what is organocatalysis. So a simple definition of organocatalysis, it's just like catalysis. Dr. Yadav has been referring to catalysis materials and other metal-based catalysis and even photocatalysis and all those things. So here I'm just picking up on something which is very simple. So when we use an organic molecule and use it as a catalyst, simple catalyst, that is what is organocatalysis. And the another condition is that there should not be any metal involved in it. So as I've written out here, is a process in which an organic molecule free from metal ion catalyzes an organic transformation. Now, this has no doubt green advantages also because it is metal-free catalysis, and metals can be may not be environmentally benign, so they can be toxic, and heavy metals, as you know. So no derivatization of the substrate is required at times, low catalyst loading, ambient reaction temperature and pressure, low solvent usage as such, and sometimes less solvent usage, no need of inert atmosphere, and no need of dry conditions. This is what we normally work when we are using organic molecules. Now, when we say organic molecules, we can call them, if we are using them as catalysts, we can call them organocatalysts. Those organic molecules which are involved in catalyzing certain transformations are referred to as organocatalysts. And there is a whole list of molecules which can be found to be acting as organocatalysts. You can just see for yourself in this limiting, I would say this is still a limiting list, a lot of molecules you're all familiar with, and they can be used as catalysts for carrying out these reactions. Okay, chemical reactions, variety of chemical reactions. There are n number of now chemical reactions that it stands today. It is a lot of reactions that can be performed using organocatalysts. I'll just take on, anyway, just to give you a brief idea before I move on to how the inspiration from nature came up for development of organocatalysts. Now, it was already being known in literature that organocatalysts are quite useful catalysts, and people were using it. But the basic turnaround came in the year 2000 when two different groups came up with this idea. One was Benjamin List at Scripps Institute, another was Macmillan's group. So they came up with two different reactions, and they showed that they can be performed with the help of an organic molecule. Macmillan was actually the one who gave the word organocatalyst for this type of transformation where organic molecules are involved. So very simply, what one needs to look at is the inspiration from nature comes if you just study the catalytic reactions that the nature is performing. And nature is known to build up large molecules from starting with very small molecules. As you can see, carbon dioxide or carbon, water, oxygen, and all those things. These are the basic things which can be utilized. Okay, although breathe out the carbon dioxide goes out, but that carbon dioxide is used by plants to make all these micromolecules and macromolecules. So another advantage of looking at enzymes and studying from enzymes is that enzymes can also induce chirality in the molecules, and the processes can be very chiral. We can get, in other words, if I say, we will be having enantioselective processes. So it's important to study the enzymes and then try to mimic them, and this is a quite challenging task to mimic enzymes by structures that mimic the action of enzyme. So all this is what organic chemists have been doing over the years and have developed a variety of organocatalysts that mimic natural processes. So I'll take a few examples of nature-inspired organocatalysis, or what people have done. Now, the first example I'm taking is a very simple protein or an enzyme which we refer to as serine protease. So serine protease are a class of enzymes that are responsible or involved in cleavage of peptide bonds of proteins. And in this serine amino acid, or the residue, serves as the catalytic or the active site or the nucleophilic site from where everything starts. So the key active sites actually is a triad which is called catalytic triad, which is composing of serine, histidine, and aspartic acid. Aspartate or aspartic acid. These groups are in the same in certain orientation line as we have just tried to show. This is just a diagrammatic representation. This is representation also which tells us how serine has an hydroxy group at its site, which is the active group, which actually interacts with the molecule. So the examples of the serine protease are numerous. Well, the one or two which I'm citing here, trypsin, chymotrypsin, and subtilisin, is something we come across quite often while we study also. These are some names which we rarely come across because these are very, very specific ones. So these are all intracellular, but this one, TADG14, is a novel extracellular serine protease that had been identified and cloned from an ovarian carcinoma. So the source is entirely different. So obviously, well, the two well-known are these digestive enzymes present in small trypsin and chymotrypsin are very well studied. So the active site of this catalytic triad, as I told you, has a serine, histidine, and aspartate in it. Now, what we can learn from this enzyme, and can we build up an organic molecule which can do the same type of transformation or can be a catalyst? So it was L. Bernardi and his group. In fact, Ricky is a senior author in this. They came up with this idea that this is how probably amide hydrolysis occurs in the serine protease. So when they analyzed, they found that there are one group is like this, the other is the serine, the other one is the histidine. So there are some hydrogen bonding sites here. So they tried to introduce urea and the thiourea unit in a molecule which actually provides a hydrogen bonding that is equivalent to this site. Then an CH2OH which acts as equivalent of a serine, a nucleophile, and this imidazole nitrogen which acts as a base. So they introduced this pyridine ring in this molecule, and they designed this molecule and tried to see if this molecule can act as a catalyst. So as it turned out, they performed a few reactions with this, and they were able to get the reactions in the positive sense. Another reaction which was actually with a similar molecule. Well, if you see this, sorry. This was there, and then if you see this molecule has also been taken care of earlier by the group of Sakai. So they have actually tried to carry out transesterification between vinyl acetate and alcohol, which is actually again mimicking of a serine, what do you call, amino acid, an enzyme, a serine enzyme, which is actually lipase. So they have tried to look at the active site of the lipase, and they came up with this, and they found that this, again, similar molecule can act as a complete mimic of that, and they could show that this reaction occurs. They could show that this transformation occurs, and it's nearly 100%. In 1 hour, this transformation occurs and transesterification is there. And anyway, they carried out some optimization studies, and they actually found that 1 mole% of this, when you have this urea unit here, that completes the reaction in 30 minutes, when you have thiourea unit here, then it takes around 1 hour or so, as shown here. So, and at low loading also they found 0.1 mole% of the loading of the catalyst. Thiourea took around 16 hours, where urea took around 4 hours for this completion of this reaction. So this unit was then referred to as a trifunctional organocatalyst, which is quite reminiscent of what I have been talking about, serine hydrolase. And it has been designed based upon the enzyme's nature, the catalytic site of the enzyme. So this actually was the first example of biometric organocatalyst creating a nucleophilic OH group, a base and an oxyanion hole. This is actually referred to as an oxyanion hole, which actually stabilizes the negatively charged oxygen here, which can catalyze and transesterification between vinyl ester and alcohol. Another example which came up, given by Liu, and they also developed a sort of a trifunctional organocatalyst. Now, if you look at this, this is the catalyst which I'm referring to. This is the figure which I have picked up from their paper itself. So they're referring to this as an acid-activated Bronsted base. So, and this is PPH2, which is actually referred to as a Lewis base, and this is OH, which is referred to as a Bronsted acid. And this binap-based phosphine catalyst have been found to be quite useful, and it has been able to. Now this has a chirality. This binaptyl has a chirality. This is the chiral framework. So they were able to get this Aza-Morita-Bellis-Hillman reaction in this case. Quite good yields, as you can see, 86 to 96%, and the enantiomeric excess would be up to 92% that they obtain. This distinctly different from that of bifunctional catalysts that require low temperature to restrict conformation or high enantioselectivity. So this was slightly a trifunctional catalyst, which was different from a bifunctional organocatalyst, because this could be one could carry out reaction at room temperature. And no doubt we can use biofunctional catalysts also at room temperature, but at times things are different. Here I introduce another enzyme, which is an aldolase. You might have come across this if you have studied glycolysis. You might have come across this. This aldolase, there are type 1 and type 2. So here I'm going to talk about enzyme. So if you look at this enzyme, it has a lysine residue, which in free NH2 group here. This in fact comes contact with compound. That is a gold donor, generates an aminium ion. Then this aminium ion gets called enamine, which undergoes reaction with this aldehyde or aldol acceptor, as you can call it. And there's a carbon-carbon bond formation with the coming of hydrolyzing it with water. The enzyme is released back, and we get this beta hydroxyketone or a carbonyl compound. So this was a basic mechanism of aldolase type 1, and it involves what is called the enamine catalysis, as it is called, because there is an intermediate formation of enamine. So this type of catalysis is referred to as enamine catalysis. So the biological process provided a template for development of small organic molecules. So it was in 2000 that Benjamin List and Babas, they came up with this idea while studying with this different amino acids, they came up with this that proline should act as a catalyst, which should provide similar results as by aldolase type 1. And they carried out a reaction in which they could find that they could get this product. I'll show you that reaction in the next, probably in the next slide. But let's have a comparison of the mechanism of the enzyme here and the proline mechanism. So just like there you have an NH2, here there's an NH. So this is secondary amine. As you can see, there's a carboxylic acid here. So it forms an aminium ion. Aminium ion gets similar to this enamine. There's enamine and an aldol acceptor. So it generates a similar sort of here. And this it releases okay, it releases the product. Okay, so let's look at this reaction, which actually was performed by this learner and Barbas. This is actually the turning point in organocatalysis. This is one of the first examples which they came up. They used acetone and different aromatic aldehydes, actually, particularly paranitro benzaldehyde, and they showed that this could be transformed in DMS. So they carried out this transformation to get this aldol addition product. And it was, again, another thing that was interesting in this was because proline is a chiral molecule, so it ensures that this hydroxy group that we have here also has a stereogenic center, is bonded to this stereogenic, a new stereogenic center in general. So it is up to 6%.
Excuse me.
Excuse me, I think there is no slide change. Your slide number 2 is still.
Let's check. You should have asked,
Yeah.
you told me earlier, so there might be something wrong.
Yeah. Now it changed.
Yeah, you should have told me that earlier.
Full screen.
And you are showing it full screen. Slide slow. Slide. You can show it in slide mode.
I don't know, but when we checked it out, it was working.
Slide show.
So I'm hearing.
You go in lower side. Yeah.
Okay, now slide happening now.
Now slide is gone.
Slides are locked. Okay, let me stop and then maybe again do it.
Sir, just share the slide as we present in the slide mode.
Let me see. Are you getting it now?
Yes, but now you have to slideshow.
I can go to slideshow. Actually, I was not able to see that. So
You go to the right side lower portion. It is a 64%.
no, when I
So you.
when I go to slideshow, it's not working.
Okay,
Without slideshow, it will work. Now it is working. Okay, I can do like this.
Yeah, okay, you continue it.
This is okay.
Okay.
So, okay. So you should have stopped me earlier, actually, when you were not able to see the slide. Okay. So I'll briefly touch upon what I left behind. Okay. What is this? Where? You can sit down here. Achha, okay. Right. Okay. So I was referring to inspiration from nature, and so this is what I started with. I was to share a few examples. Serine protease I shared with you. It has a triad of serine, histidine, and aspartate, and it has trypsin and chymotrypsin present in it. I mean, obviously, these are the serine protease enzymes. So what people learn from this, they try to develop this tri-functional catalyst with the pyridine unit and the CS2OH and this urea and thiourea unit, and they could get a similar sort of transformation that could be obtained from serine-based enzymes. And it also mimics the serine hydrolase, which we refer to as lipase. Okay, so then this is what esterification reaction that they have performed. I'm just in short repeating everything just to familiarize you with the slide. Then I came up with this another trifunctional catalyst which has been developed by Liu and others. This is a molecule like this, and the chirality is in the part of this unit that is binaphthyl, and it has a loose basic PPS2 or phosphine unit, and then there's an NH which actually acid activated Bronsted base, and then this is the Bronsted acid which they use, and they have been able to get Aza-Morita-Baylis element reaction very conveniently using this and in high enantioselectivity and high yields. So this was the beauty of designing these trifunctional catalysts. Then this is an important one which I was referring to actually when probably you got to know that something is wrong with the slides. Well, I was talking about aldolase 1, which is enamine catalysis. And aldolase 1, the mechanism is shown in this part where lysine residue, which and an iminium ion is generated, and this iminium ion then this enamine is this carbon bond formation occurs as this is how it is. Finally, it is released, and this aldol addition product is formed. The same could be mimicked with the help of a simple amino acid that is proline, and you can see quite similarity with the intermediate steps which are involved in proline also. And this was developed by Benjamin List and Barbas Cripps in 2000. Reference I was speaking to. So this is they could get up to 96% enantiomeric cis in the DMS. So was a solvent that they use, room temperature they could get, and the mechanism is again shown here. But anyway, we have already talked about this. Now the question was, we were looking at this reaction and we were very much attracted towards this concept of using a proline for transformation. Or making carbon-carbon bond, and that too generating chirality in the product molecule. So we thought of, at that time, we were also thinking of how can we perform a similar reaction in water, because proline should be soluble in water, and so can we use proline in water? But in fact, we were not able to get a very good product in that sense that when we were using this 5 equivalents of this acetone and this paranitrobenzaldehyde and proline, we were getting racemic mixture. We were not getting an optically active product. So that was our situation, and this was the yield when we were using how much amount. So the other component had a greater role to play when we were doing this transformation. Time, obviously. But however, we found that there was no reaction in this. So our objective of getting this reaction in water as an asymmetric carbon-carbon bond formation was not successful. So we shifted to we said, okay, let's drop this out if this is not working, and then try to see if pyrrolidine works in water. And it was interesting to see that pyrrolidine affects this reaction in very high yields in less than 5 minutes. I would say actually it is just adding and extracting. That is all. I am giving 5 minutes, but I think reaction was over by the time we run the TLC. The reaction was over. So we have to stop this reaction, and yield was very good. So this was interesting, although the product formed was racemic in then. But our basic idea was to go around and develop a reaction where we can get chirality. So we went about on a like drawing table, as you can call it. We tried to look at how we can do this, how can that be involved. So, and the other idea was to do the reaction in water also. So how we can achieve that. So we thought of having a group here which is not hydrophilic, like as in this carboxylic acid, but hydrophobic. And it should also have some hydrogen bonding unit present in it. And it should also provide some steric crowding. This was our basic premise for developing this sort of a catalyst. And we went along and we could get we got from literature also that iminium ion catalysis becomes more effective as the base strength of the amine catalysis decrease. So this was Spencer, we came across from literature, and we developed our hypothesis. Let us try to add an acid to this unit and make a prolinamide where we have an NH which can be hydrogen bonding and a hydrophobic group here, and have an acid along with some acid and see how the things go. So actually this NH should be in the form of a salt. So when we did this reaction, I'm just giving a brief of this. These are the few catalysts. No doubt we tried many other catalysts in this. So these catalysts was found. What we got the product, that is the aldol addition product, in 26 hours, the enantiomeric excess was 45%. So this was one catalyst which was giving us the best results. And similarly, we had another, we tried to introduce still another CS2CH3 at this very position, but we found it also gives 46% in 8 hours. So that's comparative. So there were not much. But anyway, this was our first found, and the two diastereomeric catalysts, SR and SS, comparing these two catalysts, differ in reactivity and selectivity. So the presence of water is necessary for enantioselective catalysis. The solvents, which DMSO, which List has used earlier, were actually slowing down the reaction rate and also lowering the enantioselective product. So in all, anyway, what we came up with was that we get a reaction in water using the catalyst which was designed in our lab, and the absolute configuration of the product that we found was similar to the one that was obtained when catalyzed by L-proline in non-aqueous media, which was reported by Benjamin List and others. So we did substrate screening, and we found that the best result we could get, these are enantioselectivities, not the yields. So 62% enantioselectivity we could get with this particular substrate, and there are other gives slightly different enantioselectivity. So not very high, but anyway, for being the first reaction in water, chiral reaction in water, or an enantioselective reaction in water, this was a good thing. Then we thought of using the cyclohexanone and using a similar idea about developing a new catalyst. So we came up, as we developed a variety of different catalysts, we came up with this catalyst, which was found to be very good as far as this transformation was concerned, where we could get enantiomeric excess up till 95%, and the yield was also 92%. Even the DR was quite comfortably good. So this catalyst came up, and then we studied the substrate scope, and it was, as you can see, most of these are in 90s and 80s. So the enantiomeric excesses for all these substrates. So it seems to be a good catalyst as far as this is concerned. Again, water was the medium of the reaction, so this was one good thing. So in other words, Dr. Yadav talked about green chemistry. So we were trying to develop a catalyst which is green, which is based on natural product, and also we are avoiding the use of any organic solvent for this type of transformation. So this was quite encouraging. And okay, as far as this aldolase model was concerned, then we looked at another enzyme, which is epoxide hydrolase. So epoxide hydrolase are one of the class of enzymes that belong to a family of hydrolases, and they specifically act on ether bonds. And structure of epoxide hydrolase is something like this, that it has a sort of, there's a catalytic site, and in the catalytic site is covered by some alpha beta hydrolase or family it is, but anyway, there's a lid of polypeptide chain which covers that, which is called the lid, and the lid comes off, and then the catalytic site is exposed. What has been observed in different epoxide hydrolysis obtained from different sources. This is from microbial sources, and this is from humans, as you can say, microsomal one probably. So the idea was that it is this enzyme is the one, as the name indicates, is involved in ring opening of the epoxide. Okay. So the role of the epoxide, just to touch upon what is the basic role of the epoxide hydrolysis in nature, it is involved in the detoxification of various xenobiotics in higher organism as well as in bacteria. In bacteria also it is helpful in degradation of several environmental pollutants. Actually, it works in conjunction with an oxidative enzyme which converts arenes to arene oxides, and this epoxide hydrolysis then hydrolyzes these intermediate epoxides and converts it into diols, and which then become more soluble and excretable. And so the cellular, it can be excreted out of the cell, and so detoxification can occur, as you can see in this small representation. So the examples of xenobiotics, other contaminants, and hydrolyzed by epoxide hydrolysis are like this. So one can try these and get a variety of these epoxides being hydrolyzed by epoxide hydrolysis. Well, the basic mechanism, as when it was studied, the extracellular structure of the enzyme, when it was studied, it was found that there are two tyrosine residues positioned in such a way that they activate the oxygen of the epoxide through hydrogen bonding. And then this aspartate is the one which is involved in attacking or having a nucleophilic attack on the epoxide ring and generating this intermediate enzyme substrate. Ester intermediate is generated, and then in the next step this is released, and this xenobiotic is then released as a diol, and the enzyme is ready to take up another epoxide, and so this transformation keeps on going. Here also, as you can see, histidine is an important component which actually activates water for delivery to this, and so that this ester hydrolysis occurs in the intermediate stage. Okay. So based upon this, Hein actually came up with the design of a molecule like this, which which mimicked tyrosines, the two tyrosines of the epoxide hydrolysis. And this is what it was. They developed this molecule, and they showed that this molecule was quite effective in amination reaction here, where amine could be added as a nucleophile onto the epoxide, and this was could be achieved in quite good yields. Later on, Braddock and others came up with a similar idea, but developed a different molecule like this. So they investigated 4,12-dihydroxy-2,2-paracyclophenediols and its para-substitute derivatives. As you can see, different derivatives they tried, and they carried out ring opening reaction of the cyclohexyl, and they were able to get now quite good yields of this, but not with all. So you can see different yields. This was probably the best, found to be the best catalyst for giving high yields for this. It took long time, 66 hours, but this was again representation of how we can mimic nature's enzymes and develop small organic molecules as catalysts. Then came up with this Kleiner and Schinner. They came up in 2006. They came up with an idea that the double hydrogen bonding sort of activation can also be achieved with urea and thiourea, and they found this thiourea to be quite useful, and they used a variety of different nucleophiles, aliphatic amines, thiophenols, phenols, and they could get these two isoisomers in different ratios. In some cases 1:1, in certain other cases 1:2, and which generates from attacking of the nucleophile at this carbon or at substituted or unsubstituted carbon, and so these two isoisomers was distributed. So this was another idea, and so we also was working at that time as I had interest in biocatalysis, so we were also working on epoxide hydrolase in our lab. At that time, microbial epoxide hydrolase was what we were using in the lab, and then we said, let us try to see if this really is true. During that time, when Senior actually published, we were also looking at this thiourea catalyst, but we were not having any results at that time, but we were just probing this if it can be useful. So anyway, what we were doing was we were trying to look at amines and this styrene oxide, and we were able to get 99:1. I mean, this was the best thing that we were where Senior was getting both the regioisomers. We were in certain cases having 99% of one regioisomer being formed. So this was best. This is what generates selectivity, and again we found the same thiourea molecule which Senior has found to be best. In our lab also we found this to be working very fine, and also we were working in solvent-free condition. There was no solvent that was added into the reaction mixture. So that was another advantage that we developed in this case. Then another you can say again from epoxide hydrolase and this study that we did, we thought of designing a peptidyl thiourea derivative. This, as you see, delivers us racemic product, but we were interested in getting asymmetric product or chiral product. So we then sat down and started thinking about how to design a catalyst that can deliver us the product which will be asymmetric or which will be chiral. So we thought of having using a polypeptide chain with turning it so that on one end we had thiourea, on the other end we had some anchor to anchor the nucleophile so that we can provide the right type of orientation, and this nucleophile is delivered only from one side. So this was our basic idea at that time, or the hypothesis were like this. Now the next one was how to what sort of peptide linkage can provide us a turn, and we came up with L-Pro, D-Pro. If they're in line, I mean one after the other, then these units were useful in providing this turn, or which is called beta turn. So all peptides which have L-Pro, D-Pro in sequence, they provide this turning point. So we tried to develop the catalyst with L-Pro, D-Pro, proline, actually L-proline and D-proline, and we devised these tripeptides and then try to see if they work in the same sense as we are. That's what we wanted. This work was done by Vikas. So we were able to successful to get this transformation in an asymmetric fashion up to the enantiomeric excess that we could get was 79%. That was the highest that we could get, and we prepared a variety of different tripeptides and peptides to see which one gives the best results. So anyway, the catalyst before you here is one which gave us the best. Maybe I have one more slide for you to tell you that how the things work. So this is the sequence of the catalyst, that is L-phenylalanine, L-proline, D-proline, OMe, that is the ester part, and so in the end this is the C-terminal end, this is the N-terminal end, and when we had the reverse of this, as we had 7A and 7K, when they were reversed, the opposite enantiomers could be obtained. So that was again the beauty, that we could get both the enantiomers of this product by choosing the type of tripeptide that tripeptide thiourea, tripeptidyl thiourea, and both the enantiomers could be obtained in this case. Again, this idea came from the study that we were doing on epoxide hydrolase. So that is what was the best thing for us, and whatsoever we thought of, we could execute it. And this is how the transition state we proposed probably looks like, because when we studied the X-ray crystal, single crystal X-ray of this molecule, we could get this configurations as R and R, and so from based on that, we proposed that this is probably how this is working, and we could get the products. And this actually got the cover picture. We were asked by the journal to give a cover picture for this, and this is the cover picture. My student Vikas, who just saw, designed it, and so it was there on this Asian of Organic Chemistry. Now coming to carbon-carbon bond formation via decarboxylative addition reactions. Let me look at what are these decarboxylative addition reactions, and where do we find them in nature. I hope all of you, if you've done biosynthesis in somewhere in your coursework, you've come across this polyketide synthase. So these are make up a polyketides. In fact, are themselves means that they have number of ketones in them. The molecules that have number of ketones, then they make a diverse biogenetic group, which the starting point is actually acetyl coenzyme A. It forms a linear chain without extensive reduction. So, and polyketide synthesis is an enzyme system. It has a number of different enzymes that constitutes this synthase, which actually is responsible for synthesizing these. And some of these biologically produced or natural products which have been produced are examples here, and they are have bioactivities also, these polyketides which are produced. So the important natural producers of polyketide metabolites, as they are referred to as, are fungi, molds, bacteria, particularly of actinomycetes family, then sea slugs and others. So what it is about, where is this decarboxylative carbon-carbon bond formation involved in polyketide synthesis? So for the synthesis of polyketides, coenzyme A has to come along if okay, it's not here anyway. So in the active site model of this also, there is a cysteine which carries the acid, this carbonyl or group. This is an ester, thioester sort of unit which is formed, and this is the malonate which comes along. Maybe I have a much broader about this in the next slide, so I'll anyway talk about this. Though these are malonic esters, thioesters, which actually are the building blocks, and coenzyme A is here, and this is delivered onto this with the decarboxylation of this malonic ester thioesters. Okay, sorry. Let me show this process in much better way in the next slide. So this is the polyketide synthase unit, which is having cysteine, which is having an histidine, and having another of these aspartame. Okay. So this is the acetyl coenzyme A comes up here. It comes and gets involved in hydrogen bonding with this unit, amino acid residue, and this cysteine picks up this acyl group from here, and once this cysteine picks up this acyl group from coenzyme A is again free. It can go back, pick up another acyl group and come back. And then coenzyme A SH, again, this is malonyl coenzyme A which comes along, and this again comes in touch with, is activated, the carbonyl of this thioester is actually activated. As a result, this loses its carboxylic acid, and this proton is also picked up by this histidine unit, and so the carbon dioxide is given off, and this is an activated enolate now, which then adds on to this acyl or ester unit. Now it is thioester unit, and carbon-carbon bond is formed, and a polyketide is then released. So this process goes on, and the building up or the building unit is for this polyketides is malonyl coenzyme A, which provides this carbonyl unit to them, and so on. it keeps on working. Okay, so this is the idea, and then looking at decarboxylative mechanism. So what we are concentrating on this portion where decarboxylation is actually occurring, and we are interested in getting these enolates which can really which can act as a nucleophile and add on to the reaction. So this decarboxylation, now I'll just take you to this decarboxylation. Their proposals are that there are three ways probably the mechanism is possible. The decarboxylation and condensation of melanine and acetyl units is possible. So this is one is the concerted decarboxylation addition. So it happens, decarboxylation happens, and then the addition happens on this carbonyl, and so it is released. The other supposition is that there is a stepwise process which occurs. First decarboxylation occurs. There is a generation of an enolate, which then probably adds on to which the addition process is separate. So there is a slight time gap in between, and so that is why it is referred to as stepwise. And then there is another proposal that it can be stepwise, but first addition and then decarboxylation. So first it is addition, so addition that is occurring here, and then decarboxylation occurs. So these are different proposals before it. But as we found in one of our studies that it is actually what you call the stepwise reaction, decarboxylation and addition reaction. So we came up with certain literature reports on this. I'll just share a few literature reports with you regarding this. And all these have been inspired from this decarboxylase enzyme, which is involved in decarboxylation of malonic esters, thioesters. So it is malonic half thioesters. Similar mimics were used as substrates also, and people have used metal based catalyst as well, as you can see here, and they have also had some chiral ligands along with them, or some ligands which do help. So initial work was based upon using metals for this type of transformation. Again, this was successful, no doubt. As you can see, there are a few examples coming from here, COSY group, and they were quite successful in showing that this transformation is feasible. But our focus was on organocatalytic, but the first organocatalytic transformation was reported by Venemer. So she showed that one could get this reaction very conveniently by using catalyst which has a thiourea, and this is the moiety which comes from cinchona alkaloids. Okay, so quite efficient. And so this is the sort of what you call transition state. They proposed that this is how probably this is activated. This portion is activated in combination with this double hydrogen bonding of the urea, the thiourea, and then decarboxylation occurs, and along with whether it is like concerted or stepwise, so it adds on to this nitroolefin, and this product is obtained, and they found that yields of up to 97% and enantiomeric excess of this, because a new stereogenic center is being generated, so it results in quite enantioselective transformation. So then other groups also came in. They came up with different sort of organocatalysts, and they could show that their organocatalysts also gave similar transformation. But in this case, there are two different organocatalysts, and there's a imine which is being used here. So imine and MHTs are they try to MAHT reaction. They try to form. And at 0 degree centigrade, and they could get quite good. With this catalyst, they could get only 79% of enantiomeric excess. That is the maximum they got. Whereas with this particular catalyst, they were able to. They have been introduced by two different groups, and they were also reported in 2007 and then 2011. So this was 97% enantiomeric excess. So this turned out to be a better catalyst. So that also During that time, another report came on isotins. We were working in this. We were working on these molecules at that time. Also, we were also exploring these reactions around that time. So, but still, results were awaited. So they came up with this, that there is a squaramide. Now, this unit is referred to as a squaramide unit, and this unit comes from the cinchona alkaloid. So they came up with that this catalyst is quite useful in carrying out this transformation. Again, decarboxylation is involved in this, and one could get this product in quite good yield and up to 92% enantiomeric excess. In certain cases, the enantiomeric excess was less also. So this Shibata came up with particular, and they showed that this could be transformed, this product could be transformed into a natural product through a sequence of reactions, and then the utility of this whole process they highlighted with the help of this. So, well, in our group, what we said, we were trying to look at this reaction, as I told you, and there are few projects that we handled here. So I'll take you one by one through these projects. So enantioselective decarboxylation of beta keto acids to acetinamine, which is catalyzed by chiral cinchona. So we were able to carry out this transformation and publish this in 2017. This work was carried out by Anita and Jasneet initially. So they worked on this. They did they synthesized different thiourea, cinchona thiourea catalyst, and squaramide, cinchona squaramide catalyst, and they found that this squaramide quinine catalyst turned out to be the best in the initial screening. Well, if you see in the initial screening, we were only getting 32% of enantiomeric excess. The enantiomeric excess was quite low, but anyway, kept on optimizing the conditions. And by screening the solvents, we came up with 1,2-dioxane, which was quite good, which enhanced the enantiomeric excess to 89%, and other ether solvents were also found to be good. In this case, methyl tertiary ether was also found to give quite good, but this was giving slightly higher. So we moved along with this dioxane, came up with, went about studying the what you call substrate scope of these derivatives, and what we found was quite good enantioselectivities. As you can see, most of them are in 90s, some of them no doubt are in 80s, or particularly one or two are in 80s. But when we went about looking at these molecules, they were giving the substrate scope as far as the keto carboxylates or beta keto carboxylates. When we went on to from phenyl to methyl, then the enantioselectivities were comparatively less. Okay. So substrate scope of different beta keto acids. Then we tried different beta keto acids. As you can see, this tried different beta keto acids, and the results was quite encouraging. We were getting quite good results, although in certain cases the enantioselectivities were low. So that is one issue regarding this, that not always you get very high selectivities. So after getting the single crystal X-ray of the product, then we proposed how probably the transition state must look like. So this was the favorable transition state, where the unfavorable transition state would be something like this. So since we're getting S configured stereogenic center here, so that means this is how probably everything is placed. This delivers the S configured one, whereas this will deliver the R configured one, which is slightly disfavored in this case. We also studied, can we do this in one pot, this reaction, because this required the generation of the acetinamines. So we tried to see how it works. Anyway, we generated acetinamine, and to the same reaction mixture, to the same pot, we in fact added our catalyst and the other reactant, that is beta keto acid, and we could get the same product in quite good yield, but the enantiomeric excesses were comparatively lower than we were working with pure systems. So the idea was simply to tell that one can also develop a one-pot reaction, and it works. The only thing was that the selectivities somehow was lower. enantioselectivity was lower of the product that we were getting. Tried to do some little bit transformation. We tried many things, but ultimately we found that we can make a thiourea out of this and see if this can be of any use. The product was quite good. So then coming to the next project, here we were again involved in methylene indolenones with beta ketoesters. So the substrate changes here. And we thought of doing this reaction and see this, although we tried many other substrates, but not getting very good results in most of the cases. Well, there were other, here again we found the same catalyst was found to be delivering the best results. And in the very first instance, we found that quinine squaramide catalyst, as shown here, which was also found to be earlier the catalyst of choice for the transformation, was also found to be good. But the solvent screening when we did, we found that CPME gave us better, what you call, enantiomer excess for the product. So we continued with this, and after the optimization of the conditions, we came up with what you call the substrate scope. And the substrate scope also, if you see, we are getting quite good results. Here we could, since we were getting two diastereomers, they were nearly into 60:40 ratio. So, but individually they were quite enantioselectively formed. As you can see, we have given the enantioselective, the enantiomer excess of both the diastereomers in these tables. Okay, so coming to this other situation. Just a minute. So we studied the other portion of substrate scope was to vary the beta ketoesters, and when we did that, we were getting more or less similar results. So there were no issues regarding this. So the of this catalyst was then established again, as we have been doing earlier. We tried to isolate the diastereomer, which was formed in larger amount, and then look at and crystallize it, and then try to look at its extracrystal structure. So this was formed in larger amount, and based on the absolute configuration that we got, we tried to show that this propose the transition state. So this is how probably it looks. So anyway, this is about how we looked at all these things. Then another substrate we tried was parazolone alkene. Now the parazolones we found was quite important units and they were part of some natural products, alkaloids, and also showed bioactivities, and there were a lot of molecules available in literatures. A few of them we are just representing here, and this is what we tried to do, and we tried to see if this works. And anyway, the reaction was working. So the question was, can we get enantiomer excess? And again, this quinine squaramide came out to be the best for this, and the enantiomer excess was only 59% in our initial screening of the reaction. So we went about the optimization. Here also we came up with 1,4-dioxane, which turns out to be best. Another thing that we observed was, if you add molecular sieves to the reaction mixture, that probably helps in removing the traces of moisture. The enantioselectivity as well as the yield was enhanced. So we performed this, became our standard procedure, and we performed. This was the optimized conditions that we used then. And we performed this reaction and studied the substrate scope. So if you look at the substrate scope, there's a large amount of molecules that we did try, and the enantiomeric excesses were not very high, so we could not get up to 90%. But anyway, this turned out to be, the yields were good, no doubt about it, but this turned out to be a good example where we could get this transformation. So scale up of the reaction was done. Here we were working with, if you see, 0.1 millimolar. Then we went up to 1 millimolar, and we found that again the yields are good, but that has some effect on the enantioselectivity of the product. So the last project in this was again we were looking at some non-conventional methods of carrying out reaction, where we were trying to do grinding reaction. So we were trying to see if this reaction can occur under grinding condition and solvent-free condition, simply mixing the two together and grinding them in porcelain mortar in presence of a catalyst. Can we get this or not? So what we found was, here we were like looking at different amines. We found that DBU was giving us the highest yield and the very best time. DBU was the catalyst which gave us very good results. So we went about, now this is not a chiral work, because initially we were trying to look at grinding, because the idea was to do that grinding. Okay. This can be done in ball and mill also, but we were not having ball and mill. We did grinding with the hand. The student did it with the hand. And in 5 minutes, this reaction could be achieved. The product could be formed. You could simply isolate the product, wash it with ammonium chloride solution, just to remove the amine catalyst that we have added. So that got removed, and the solid product could be isolated, and that was found to be quite pure. So there was no problem. We don't have to go for column chromatography in this case. So it was like column chromatography sort of a thing that we got. So we also try to do one pot reaction, as we have done in earlier cases. So here also we were successful, and the yields were quite good. So 1 3-component reaction, 1 pot 3-component reaction, where this intermediate is also generated, and we believe one for addition, reduction, cyclization. And then we also tried this transformation, and we could get this pyroxidol after this had been formed. This was also then tried 1 pot, and we could get this. That means we were grinding actually. Okay, porcelain mortar. After you have done this job, then you wash this, dry it, remove DBU from it by washing it with ammonium chloride, and then as it dries up, you add a little bit of methanol, and then sodium borohydride, and just do the grinding again, and you end up getting this product. So this was a really good job which we could attain. So these were some of the projects that we handled in decarboxylation. Some of the other things I've shared with you in the earlier is, so what we have come out with, that the natural products actually were the ones which slightly modified natural products came out to be a very good catalyst for our case. And Syncona based these catalysts were the ones which were very good. I'm not connected. Okay. There's something wrong. Sorry, there's something wrong. Okay, just a wait. You should have told me that it's not coming out. You're not able to see the slides probably.
Yes, there is some.
Yeah, there were some network issues. Okay.
Please continue.
Let me again join the meeting. Okay, I have to again join the meeting. Okay, okay. So, just a minute. It's slowing down a bit. I don't know what's the reason. Okay. Maybe be some issue with the connectivity part. Okay, there's no need for this. So let me, if I can again share. Can you see it now?
Yes, now it has been shared.
So I don't know when it stopped coming. So because I'm using my and the desktop for that. So you should have stopped me and told me about it. But anyway, I'm just confused. Should I go and conclude, or do you want me to tell you? Okay. So anyway, what I wanted to convey to through this lecture is that you can use, you can think of using, looking at enzymes and try to mimic them, and also in the process, for mimicking them, one can use natural products, and or one can modify the natural products to just mimic the action of the enzymes. And in this case, we have, as I've showed you in the decarboxylation part, we have used cinchona alkaloids, and we have modified cinchona alkaloids to generate what are called cinchona-based ceramides, and they were found to be excellent catalysts in all these cases. And we do expect that organic chemists and in journal chemists can look at nature, watch nature closely, and then try to develop some similar processes which nature is doing, and I'm hopeful that most of you would be successful in doing it. So this is what I had for you today. I do acknowledge grants that I've been getting, the funding that I've been getting from Council of Scientific and Industrial Research, the Science Engineering Research Board, and UGC for all my research. And my students have been getting their fellowships from them. And so thanks to the funding agencies of Government of India. That's why we have been able to produce creativity in our work. It's all based upon what money that we get. So this was my research group, which was involved when decarboxylation was done. Anyway, the work was initiated by Jasmeet and then carried over by Anita. They were the two who was involved, and all others are now, they're more or less left my group. And there's this young guy you can see he's from JNK. He's in, I think, Government College, Bandipura. Bandipura, he's a lecturer, assistant professor there now. So he was a postdoc with me. These are the other members, and this is our beautiful campus, as you can see this. Thank you for your attention, and sorry for all those small issues that came along when I was presenting. Okay. Thank you very much.
Thank you, sir. On behalf of Patron, Convener and Organizing Committee, I'm very pleased and thankful to you for your delightful, wonderful and knowledgeable presentation. It was really interesting and informative talk. Personally, your presence means a lot to me. Hopefully, all the attendees have a great time for getting such wonderful and informative session from your side. I further extend my thanks to you for giving your more precious time and sharing your knowledge and experience. Thank you so much, sir.
Thank you, Nasib. But if there
and I will be joining you soon in the project at Guru Nanak Dev University, but due to COVID lockdown, it is little bit pending process. So, if anyone has any questions or query, please write in the chat box. Any questions? Please write in the chat box.
Vikas Yadav says, thank you, sir. And you're welcome, Vikas Yadav. Then Lakshman Durya Swami again says, thank you for your excellent presentation. Anyway, thank you for being there and in a good audience, being a good audience, but hope that you can also have some questions.
Okay, in the end, I would like to express my thanks to all the attendees for your presence here. Our morning session is over here. Our next session, that is afternoon session, will start at 2 p.m. Kindly join us at 2 p.m. sharply. Thanks a lot.
Okay. Thank you. Bye-bye. Bye-bye. It was great.